And we're back. I guess I didn't manage to scare you guys off, huh? Oh, I cracked myself up. Okay. So, just to recap, in case it's been a while since you watched the last video, we covered reduction and oxidation reactions in the last video. Here we're going to look at exergonic and endergonic reactions and isomerization reactions. So, let's go to our exergonic and endergonic reactions. Alright, so very simply, an exergonic reaction releases heat. An endergonic reaction absorbs heat or energy. Either one. Think exer, like X means former, right? So, or outside. So you are putting energy outside of yourself. Endo means inside, so you're taking energy inside of yourself. And a reaction can be both a reduction reaction, so you can get electrons, and you can get their energy too. So a reaction can be both reduction and endergonic. Here is what we call energy curves down here. These are called energy curves. These track the energy of molecules during a chemical reaction. And this is an energy curve right here for an exergonic reaction. Okay, an exergonic reaction. As an example of an exergonic reaction, this is actually one that we used before. We're going to take sucrose. We're going to break it apart into glucose and fructose. Into glucose and fructose. And hold on one sec. Hold on one sec, guys. Hey, Jake. Can you take a look at the girls? I think Jack's bullying Tally. Sorry, I got a pair of rats, and they are not behaving very well right now. Okay. So, exergonic and endergonic reactions. Where were we? Oh yeah, sucrose into glucose and fructose. So this is a hydrolysis reaction. If we were to proceed with this, this would be a catabolic pathway. If you remember that, cats destroy things. Okay. So sucrose has, relatively speaking, has pretty high energy. Sucrose is the molecule that's going into this reaction. We call any molecule that's going into reaction a reactant. Alright. Any molecule or molecules that are coming out are called the products. So products are what you produce. With sucrose, when we hydrolyze it, we are releasing energy from that bond, from the glycosidic bond in sucrose. And that's the energy that's released right here, represented by this gray arrow. And the enzyme sucrase is the one that hydrolyzes it. Now you'll notice that there's this little bump right here. That little bump has the label activation energy. That is the energy required to get a chemical reaction going. It's the energy required to get a chemical reaction going. Is Jack being a jerk? No? They were totally squeaking and tumbling around the cage. Okay, I just wanted to make sure. Sorry about that. Okay, activation energy. This activation energy, by the way, is why all the sucrose molecules on Earth are not currently breaking apart into glucose and fructose. As a molecule, you would ideally like to be in your lowest energy state, right? Molecules like to be lazy, they like to be spread out. So sucrose would like to exist as glucose and fructose, or even better, just plain old carbon dioxide. But it has to get over this activation energy barrier. And by the way, activation energy is also sometimes called energy of activation. And this is the reason why, again, our sucrose just doesn't spontaneously devolve into simpler sugars. And sucrase is the enzyme that gets sucrose over that activation energy bump. So as an example, if I put a beaker of sugar, right, just on the table in front of you, and I asked you how long it would take that sugar, that sucrose, to break down into glucose and fructose, you'd probably be saying, a couple thousand years, right? And you'd be pretty close to correct. More like a couple million years, but, you get the idea. On the other hand, if we added the enzyme sucrase to it, that sucrase would chew through that sugar in no time, man. All that sugar in there would be gone and be broken down into glucose and fructose within minutes. So enzymes will often speed up chemical reactions by lowering or providing the bump to get over that activation energy. All right. Now with endergonic reactions, on the other hand, you may start off with a molecule that's fairly low energy. Let's take carbon dioxide, which is very low energy. Not even fairly low, but very low. And let's say we're going to do photosynthesis. We're going to build that carbon dioxide up into glucose, up into sugar. So photosynthesis takes CO2 and anabolizes it, builds it into glucose, into sugar. Okay? So that's going to take a lot of energy to do that because we are building. This is a huge anabolic pathway. We have to go up this huge bump, this huge energy curve right there. And what provides that energy is, well, it's actually several different enzymes, but we're just going to pretend it's one here for the sake of simplicity. It's enzymes that supply that energy. But very often in these reactions that require a ton of effort, you need some additional support, some additional energy in the form of a molecule, another molecule called ATP. We're going to come back to ATP here. Okay? So notice here we also have an activation energy. The analogy I like to use here with this activation energy is, imagine a chemical reaction is like running. It's like sprinting at top speed, right? And you're sprinting, and you're in a race and you reach the finish line, but you can't just stop dead, right? That's kind of what's happening in a chemical reaction. These molecules are racing around, exchanging electrons, changing up their bonds. They can't just stop dead. It takes them a little bit to actually come to a halt. That's what that bump right here is. It's kind of that time it takes for them to come to a halt. Over here, the opposite side with an exergonic reaction, that bump is what it takes to, to like start a ball rolling down the hill. Think of it this way, right? There's a ball on a hill, and you have to give it a little push to get it rolling. That little push is kind of like the activation energy for an exergonic reaction. This YouTube link down here, by the way, this is from a show called Mythbusters. I don't know if you guys have ever heard of it, but they are demonstrating a massively exergonic reaction where chemical potential energy is converted to heat. They're using a chemical called thermite to try and melt a car. It's pretty cool. So take a watch. Okay. One particular type of exergonic and endergonic reactions that I want to discuss was something called a phosphorylation reaction. A phosphorylation reaction. And phosphorylation probably makes you think of phosphate. And remember, a phosphate, it's one of our functional groups. It looks like that. It's just a PO4. Oops. So phosphates are very, very high energy, right? There's lots of chemical potential energy in that phosphorus and oxygen bond. And a phosphorylation reaction is adding a phosphate group to a molecule, We can add phosphate groups to glucose. We can phosphorylate glucose. We can add phosphate groups to proteins. We can phosphorylate proteins. We can phosphorylate lipids. We can phosphorylate damn near everything that we want to. All right. Now, actually creating that phosphate bond, attaching the phosphate group to a molecule though, right, you're building a chemical bond that requires energy to do that. So the process of phosphorylation itself, adding a phosphate group, itself is actually an endergonic reaction. It requires energy. But once you've added that phosphate group to a molecule, you've increased that molecule's chemical potential energy. Because again, there's lots of energy in that phosphate-oxygen bond. Okay, so phosphorylation reactions increase a molecule's chemical potential energy. So right here, we have a molecule. Remember, there's CH3 over here. There's a carbon right here. We can replace that that minus right there with just an H, so we can think of this as an OH. Okay. With a phosphorylation reaction, we will add on a phosphate group. In this case, we're going to add the phosphate group right onto that OH and take off the H. This is not always the case. Sometimes we might add it onto this O right here, but just for this example, we're going to add it onto this H and take off the H. Here we've added the phosphate group, and we've increased this molecule's chemical potential energy. Now, here's the thing. Just like we needed electron storage, remember we had our NADH and FADH2 as our electron storage. Remember those guys? Oops. We have to have a chemical potential energy storage molecule. Okay? And that molecule is this guy called ATP. ATP stands for adenosine triphosphate. Adenosine triphosphate. And ATP, the whole point of ATP is just to have phosphate groups. It has 3 phosphate groups. So here's what ATP looks like. And ATP is a nucleotide, right? Just like our NADH and FADH2 are nucleotides, ATP is a nucleotide. We've taken nucleotides and reused them for other purposes in the cell. I don't know if you guys remember, but way back when, we said that one of the functions of nucleotides was not only to act as DNA and RNA, but also for energy storage. Here it is. Okay. So again, here's our nitrogenous base. Here's our 5-carbon sugar, ribose in this case. And instead of 1 phosphate group, we have 3. So ATP stores energy just by being phosphorylated. Just kind of like a bank account stores money by having money deposited. And ATP can only really hold 3 phosphate groups. I don't know why we can't go up to 4. It probably has something to do with resonance structures in organic chemistry, but I'm not sure. But so we're just stuck with 3. And that's why it's called triphosphate, for 3. So there's lots of chemical potential energy in those 3 phosphate groups. Now, just like a bank account, just like a bank account can be empty, ATP can be empty in terms of chemical potential energy. So here's the bank account of a millionaire right here. 3 phosphate groups, lots of chemical potential energy. Here's the bank account of someone who's not a millionaire. Here we just have 2 phosphate groups. We've ripped off one of them. And by the way, removing a phosphate group, right, undoing phosphorylation, is called hydrolysis. And yes, we do need a water molecule for that. I know you're scratching your head and you're probably going, well, why the hell don't we call it dephosphorylation? I don't know why. I'm sorry, we don't. We call it hydrolysis. So anyways, when you hydrolyze off this phosphate group at the very end, when you hydrolyze off that phosphate group, you are releasing the energy that was formerly stored in this bond. You're releasing that chemical potential energy. And you can use that energy to do other stuff like active transport. A lot of our ATP actually goes to active transport. This molecule right here only has two phosphate groups, so we call it adenosine diphosphate. We can go even further. We can rip off another phosphate group, hydrolyze off another phosphate group, and then we have AMP, adenosine monophosphate. So this is when your bank account is empty, right? And you're starting to get overdraft charges and bounce checks. And what's that shitty thing that banks do where they charge you if you have below a certain amount of money in your account, right? So you're at that level. Anyways. Alright, so we don't really use AMP for too much. It's too low energy to be used for energy storage. Okay. So, we're really going to focus on ATP and ADP. D with a dog. Alright, ADP is in dog. I was totally thinking of a different D, but we're not going there, okay? So ATP has more chemical potential energy. When we hydrolyze ATP, we're removing that phosphate group. We're getting AD with a dog P. This is less chemical potential energy. Now we can just rephosphorylate it, add another phosphate group, increase its chemical potential energy again, deposit more money in that bank account. So, why do we do this? Well, think about it this way, okay? Let's go back to eating breakfast. So say you ate breakfast. You ate breakfast, right? Okay, good. And you took in a lot of glucose with that breakfast. You're going to catabolize that glucose through several steps, several catabolic steps. And ultimately, you're going to turn that glucose all the way into carbon dioxide. And we're going to extract electrons and a whole bunch of other stuff from that too. There's a lot of stuff that's going to go on the side. We're going to ignore that for now. Alright, what we're really going to focus on is the extraction of energy. So we are extracting chemical potential energy from glucose. Chemical potential energy from glucose. Chemical pot, okay. Alright, I'm done being immature. Okay, we're extracting chemical potential energy from glucose. And we are taking ADP, using that chemical potential energy to convert that ADP into ATP. Because remember, conversion of ADP into ATP takes energy. So, damn it, what did I do there? I'm totally lost. There we go. Okay. So we're feeding that chemical potential energy in. So you are breaking down your food, you are catabolizing your food, not only to get electrons from that food, but also to make ATP. And ATP is like money in the bank. Alright? So we can then use all of our ATP to do stuff like active transport. So that ATP we're then going to store for a while. And when I say for a while, I mean like a couple seconds. You use up most of the ATP in your muscles by standing up. So I really do mean a couple seconds here. We're going to convert that ATP, let's do green, back into ADP by doing cellular work. And that cellular work, remember work is movement. Active transport, etc. You are constantly cycling through your ATP and ADP molecules in your cell. You have a reservoir of them. Your cells each cell uses about 10 million ATP molecules a second and converts it into ADP. If you didn't have ATP, if you ran out of it, you'd be dead. That's actually a pretty good definition of death, no more ATP. So, we talked about cellular work. One of the main ways that ATP allows us to do cellular work is by changing protein shape. This is really, really, really important to remember. There will be quiz questions, there will be test questions, and we will get it wrong. So, the main function of ATP is to change protein shape. It's to change protein shape. Jake, what's the main function of ATP? He can't hear me. Oh, there we go. Change protein shape. I don't know if you heard him. All right. Please remember this now and forever, okay? Change protein shape, change protein shape. Here's an example. Here's a pump protein, active transport, right? Right now it's closed. Pump proteins are found in the cell membrane. We just see the protein here. We don't see the cell membrane. But if it was there, there would be. Okay, there's our cell membrane. What we're going to do is we're going to take an ATP molecule that we made as a result of breaking down our breakfast. We are going to hydrolyze it into ADP and take that third phosphate. The third phosphate is often called PI, stands for inorganic phosphate. Inorganic because it's no longer attached to carbon. And remember, organic molecules are carbon-based molecules. So we take that phosphate, or inorganic phosphate, whatever you want to call it, and we stick it onto the protein. We actually physically attach it to the protein. We covalently bond it to the protein right about there. Stick it right there. Okay? And that phosphate will cause the protein to change shape. And because we changed the shape, we changed the function. And that protein pump goes from closed to open. Now it will pump molecules through the cell membrane. So why does that magical phosphate group and the transfer of that phosphate group do that? Well, remember, phosphate has lots and lots of oxygen around it. Lots of electrons hanging around there. So, if you add this big old phosphate with all of its electrons to a delicate little protein with its hydrogen bonds and ionic bonds and all that, you're gonna really screw up those hydrogen bonds. You're gonna change that pattern of hydrogen bonding. You're gonna change the pattern of ionic bonding. You're gonna change all those intermolecular forces in protein shape. And so you're gonna change the protein's overall shape. Phosphorylating a protein, adding a phosphate group to a protein, can dramatically change a protein shape. And so the main way we use ATP is to phosphorylate proteins and change their shape and change their function. Here's a really good example, and this is really hard to see right now. There's a lot of information that I'm purposely leaving out to not totally overwhelm us. But this is actually an image of how your muscles use ATP. So your muscle cells use ATP in order to change their protein shape and in order to contract. So right here, this is inside a muscle cell. These are the little cytoskeleton fibers inside a muscle cell. It's actually a microfilament that I'm pointing to right there. This right here is a little lever. It's a little protein lever. It's called myosin. The microfilament's called actin in this case. The little star right there, that's ATP. Okay? So when the myosin little lever attaches to the cytoskeleton, it has ADP attached to it. When it cocks backwards, and that's the technical term, it cocks. Alright, when the little lever moves backwards, it's sliding the cytoskeleton backwards, and it's causing the overall shape of the muscle cell to shorten, to contract. It's losing the phosphate group. And then when it detaches in order to reattach itself to that actin filament, it acquires ATP. So your muscle cells use a combination of ATP, ADP, and the phosphate, the release of the phosphate in this case instead of the addition, but it does the same thing, it just changes the protein shape, to cause your muscle cells to change the shape of their cytoskeleton so your muscles contract. So go ahead and flex your arm, right? Flex your arm, pretend you're at a bodybuilding show or something, right? Well, you've just used about a couple trillion ATP molecules right there. We will do a little bit more of this in class. I'm going to have you guys flex your muscles in class. So, by the way, if you get up and you go get a drink right now, you're going to use up about 90% of the ATP molecules in your legs. And then you're going to start relying on glycogen. So our last type of chemical reaction that we're going to look at, this is the easiest one. This is called isomerization. And remember, in chemistry, an isomer is simply a molecule that has the same number and type of atoms, but different arrangement of those atoms. One of the cool things that enzymes can do is they can flip around those bonds, they can rearrange the chemical bonds, they can make one molecule into an isomer of itself. So here we have a molecule called fructose 6-phosphate. We are converting fructose 6-phosphate into glucose 6-phosphate. And I know these words are scary. It makes sense though. Hold on, let me show you why. Remember this molecule right here, this is fructose. This is one of our carbohydrates. It's the one that tastes sweet. Here's carbon number 1, carbon number 2, 3, 4, 5, carbon number 6 right there. There's a phosphate group on carbon number 6. So fructose 6-phosphate. That's why it's called fructose 6-phosphate. Glucose 6-phosphate on the other hand, 1, 2, 3, 4, 5, 6. There's a phosphate group on the 6th carbon of glucose. Alright, so isomerization reactions kind of seem sort of useless. A lot of times we're just rearranging a molecule to make it usable by the next enzyme in a metabolic pathway. And we'll see a couple examples of this later on. By the way, the name of these enzymes, if you look at them, isomerase. It's right in the name, isn't it? Isomerase. Okay, so here's our type of reactions in summary. Our first type that we looked at are redox reactions. We use NADH and FADH2 to store electrons from redox reactions. Our second major type of reaction that we looked at are exergonic and endergonic reactions. Okay, and we looked at one specific type of exergonic and endergonic reactions called phosphorylation. And we use ATP as the energy storage molecule using phosphorylation to transfer energy from exergonic to endergonic reactions and vice versa. And that phosphate group on ATP very often is used to change protein shape later on. Isomerization reactions can be used to rearrange a molecule so the next enzyme in a pathway can use it. So it prepares a molecule for use. One final really cool example here. All right. So this is what happens in your eyes. So in your eye, let me draw an eyeball. Here's the cornea of the eyeball. So this is if you're looking at the side of your head. And anything that you see, the light from anything that you see is coming in the front. Okay, it's going to hit the back of your eyeball like that. All right. Back here, this is your brain. Oh, it's a shitty brain. Hold on. There we go. A little better. Not much better, but a little better. Okay. And there's your forehead right there. Okay? You get the idea. So on the back of the eyeball, we have a very, very neuron-rich area called the retina. And in your retina, you have cells called rods and cones. They are cell types, these rods and cones. Here's a rod cell right here. Let me draw it out. It actually kind of looks like a little rod. Here's the nucleus of the rod cell. I actually did a pretty darn good job in my rod cell. So embedded in the rod cell, we have these transmembrane proteins. These transmembrane proteins are involved in cell signaling. They're involved in signal transduction. They are called, do I have the name here? Yeah. They are called rhodopsins. These transmembrane proteins are called rhodopsins. And these rhodopsins are sensitive to light. So when light hits these rhodopsins, it triggers a chemical reaction. And here's that chemical reaction. Here's a rhodopsin right here. And in the middle of that rhodopsin, we have a molecule, see that little guy in purple right there, called retinal. It has a long, complicated name. It's called 11-cis retinal. Don't worry about that. When light hits that rhodopsin and the retinal within the rhodopsin, it causes that retinal to change shape. That's an isomerization reaction. So when light hits your eyes, that's an isomerization reaction. That's what occurs. Now this form of retinal right here, the straight form, actually triggers this rhodopsin to send a signal up the length of this rhodopsin cell through the back of the retina and then to the brain. Now once we have a straight retinal like this though, it can't be reused. We have to discard it and we have to make a new retinal molecule. Our rhodopsin's still there, our protein's still there, but that little retinal molecule in the rhodopsin we have to regenerate. We regenerate it by taking vitamin A and oxidizing the vitamin A into retinal. This is why we need vitamin A in our diet, and we need a pretty constant supply of vitamin A so we can supply our rhodopsin transmembrane proteins with their retinol so we can see. Without retinol, we have no sight. Without vitamin A, no sight. So the most common form of blindness, I think in the world, or at least one of the most common forms of blindness in the world, is actually caused by a lack of vitamin A in the diet. Very, very, very easily cured simply by eating something with vitamin A. Alright, so we're going to call it a day on this stuff, and I will pick up with you guys when I see you next in class. Thanks for sticking with me, as always.